Welcome to the second lecture of the series. This is still sort of going to be an overview, and we're also going to recap just for a little while what we did in the first lecture. So, recap, what did we talk about in the last lecture? Well, we briefly gave an overview of how a nerve cell works. So, let's say we have a sensory neuron. You can see this familiar structure over here. And on one end you have information, and on the other end you have the brain. So we said in the previous lecture that whenever an information is sensed, detected, or perceived, it triggers an action potential in this area. So let's just remind ourselves what an action potential means. It means a sudden change in membrane potential. So a nerve at its resting state will have negative 70 millivolts as its membrane potential. However, the triggering of an action potential means that that membrane potential jumps to around a 60 to 70 millivolt value. And I gave a value of 100 millivolts, I think it was at the last lecture. I found out that that was a bit inaccurate. It's a bit too high. This is the normal usual range that an action potential triggers, right? It triggers this increase till 60 to 70. 100 is just a bit higher than normal. And so we have this sort of conversion from information into action potential. So the problem is this action potential is only localized to this one specific area where the information is retrieved. So everywhere else, here, they will have negative 70 millivolts, the normal, usual resting state. So the problem now is, since the information essentially translates to the action potential, how do we make sure that the brain is able to detect and read and interpret this action potential? Because right now the brain can't detect the action potential. It's all the way over here, right? We have to make it so that on this end of the nerve, we also have a 60 to 70 millivolt membrane potential. So how do we copy the signal from over here to over here? In the previous lecture, we proposed diffusion. So the way an action potential is generated is, well, we went from negative 70 millivolts to around 70 millivolts, and that means that the inside of the cell has turned more positive than the outside of the cell, and that can be caused by, say, some positive ions from outside moving to the inside of the cell. And so we thought, well, maybe these positive ions via diffusion can just move out to the different parts of the cell, and therefore the other parts of the cell will also be positive. Well, that doesn't really work because the positive charges just thin out as it spreads out. So by the time we reach over here, the signal has died down significantly, and it's a pretty ineffective way. So that's where we left off last time. I should also mention that this mechanism, we haven't uncovered what it is yet, but this mechanism is universal throughout all nerve cells. So I've been constantly using sensory nerve cells as an example, and I don't want you to think that this mechanism is specific to sensory nerve cells. It can also happen in motor neurons as well. So if we have a motor neuron over here, and then this connects to the muscle. So if we have an action potential at like this area, it will travel along the axon via the same mechanism that is used in the sensory nerve cells. And it will go to this end, and it's the same principle essentially. So the brain is detecting the action potential. This time the muscle is detecting the action potential, and it reacts accordingly. So when an action potential is detected, the muscle, what it will do is it will contract. And so some of you may be thinking, how is the action potential in the motor neuron triggered? Because over here, this is triggered by information or external stimulus. This is what leads to the triggering of the action potential. What happens over here? So the action potential actually comes this time from the brain. So the brain has neurons, say this is one of them. And what happens is, after the brain has obtained information and decided what to do in response to that information, it will trigger an action potential towards this nerve cell over here. And the action potential from this nerve cell moves across the axon, towards the axon terminal, and then it will jump from the axon terminal to the motor neuron. So besides the axonal movement of the action potential, we also have a cell-to-cell movement of the action potential. So there are two different ways in which an action potential can travel in our body. So for the first part of the lecture series, we want to be focusing on this aspect only, and we will look at this later on. So before we talk about the movement of the action potential, I think it's important to understand as well how this action potential is generated. We know how it is generated, but we only have a very short, brief description of it. Just that positive ions from the outside goes in, and it causes the membrane potential to go up. And this change from negative 70 to 70 is what is known as the action potential. Right? That's our short description, but now we want to delve into the finer details. The action potential, this has something to do with movement of ions, and movement of ions has something to do with the plasma membrane, because the plasma membrane, it's this barrier that determines whether or not stuff can go in or stuff can go out. So it dictates the movement of ions. So to understand this, we need to understand this, which is exactly what we'll be doing soon enough. So right now I want to direct your attention to this table over here. It is a table which gives us the internal concentration. Let me pick a better color. The internal concentration and the external concentration of several common significant ions. So internal concentration, of course, is the concentration of the ion inside of the cell, and the external concentration is the concentration of the ion outside of the cell. So for example, we can see that potassium is quite abundant inside of the cell, but not so much outside of the cell. Sodium, on the other hand, is abundant outside of the cell, but not so much inside of the cell. Chlorine is abundant outside of the cell, but not inside of the cell. So A- over here, that's a group term. It is, instead of one single thing, it refers to a group of different anions. And these include sulfates, phosphates, and negatively charged amino acids, and some other stuff. And yeah, that will of course be inside of the cell, right? You want these nutrients, these essential things that the cell needs, you want them to be inside of the cell. And there will be nothing pretty much outside of the cell. So we can see that potassium is the major driving force of positivity inside of the cell. And that positivity is countered by the A- group. While outside of the cell, the positivity is majorly driven by the abundance of sodium, and that positivity is counteracted by the abundance of chlorine outside of the cell as well. So right now everything is balanced out. However, if we were to create an imbalance, say for example we want to move some chlorine from the outside to the inside, then that will make the outside of the cell more positive and the inside of the cell more negative. And that's going to lead to a decrease in membrane potential. So this table is pretty important. It tells us the answer to the following question. Which ion is responsible for the generation of an action potential? Right, and this table can give us the answer to that, surprisingly, even though it doesn't look like much information. As well, we also have a yes or no, depending on whether or not this substance over here can pass through the plasma membrane or not. So potassium can pass through the plasma membrane, chlorine can pass through the plasma membrane, but A- cannot pass through this plasma membrane, and so on and so forth. So we also have that information as well. So where were we? Yes, the question. Let's think about the question a bit more. So what is an action potential? Well, it is the change of membrane potential from negative 70 millivolts to about 60 to 70 millivolts. So that is what an action potential is. And this means that the inside of the cell has become more positive. We went from a negative value to a positive value. So let's try to look at each of the possibilities, right? Let's start with potassium. In what direction should potassium move so that the inside of the cell becomes more positive? Since potassium itself is positive, it means that we need to take potassium from the outside to the inside. But hang on a moment, we don't have a lot of potassium outside of the cell, so I don't think that's going to work out. Alright, let's move on to the next possibility. Chlorine. Right, so in which direction should chlorine move in order for the cell to become more positive? Well, chlorine is negative, so we want to get that negative charge out of the cell to make the cell more positive. So just get chlorine out of the cell. But again, we run into the same problem. We don't have a lot of chlorine inside of the cell to begin with, so that's not going to produce much of an effect. So chlorine is a no-go as well. And that leaves us with A minus. So A minus, we have a bunch of it inside of the cell, but we can't afford to release it outwards. It's not permeable, so it can't pass across anyway. And even if this N were to turn into a Y, this is going to be very counteractive because you're throwing a bunch of nutrients out for the sake of generating an action potential. You don't want your nerve cells to starve every time an action potential needs to be generated. You can see how this is very bad. So A minus is also a no-go as well. These are essential nutrients. We want them in at all costs. So that leaves us with sodium. So in which direction should sodium move in order to make the cell more positive on the inside? Well, sodium has to move from the outside to the inside. Okay, but there's a problem. It's not permeable, but the star is here for a reason. So since this is the only other option left, right? So there must be some mechanism, some way that the cell can bypass this issue over here. What if the permeability to sodium only changes whenever an action potential needs to be generated? How would that mechanism work exactly? Well, something has to change about the plasma membrane, right? The plasma membrane, as you can see over here, it dictates the movement of the ions. So some structural feature of the plasma membrane should change to allow for this change in permeability. So let's draw the plasma membrane. And what are some of the structural features of plasma membranes? Well, there are membrane proteins. Some of them include protein channels. I think we're getting somewhere over here. So what if there is a protein channel that accepts sodium? And what if there exists some sort of mechanism or regulation that the cell has in order to control whether the gates of this channel are open or closed? So that's pretty interesting, right? This will just be a sneak peek of what is to come. So I think we'll leave it off at there. You can treat this as a sort of continuation to the first lecture. This is just still an overview of the entire process. And in future videos, I don't know exactly when, we'll start to take a look at these details in a much higher magnification. But for now, this is just an overview. And I still don't know how I'm gonna structure this yet. I'm doing this along the way, not pre-planned. So we'll see. Okay, that's it. Goodbye.